Recent developments in antiviral drug research, particularly from the University of California, San Francisco, UCSF, represent an exciting leap forward in combating viral diseases, not just for COVID-19, but for future pandemics as well. Researchers have identified novel drug candidates that show remarkable potential in treating SARS-CoV-2, the virus responsible for COVID-19, and even MERS, Middle East Respiratory Syndrome, another deadly coronavirus. These compounds have outperformed the widely known Paxlovid, the antiviral treatment that gained prominence during the height of the COVID-19 pandemic in preclinical testing. The significance of this breakthrough cannot be overstated, as it represents a highly promising advancement in antiviral treatment that could reshape our approach to future global health crises. The new antiviral candidates, AVI 4516 and AVI 4773, have shown incredible potency in stopping the virus from replicating by targeting a critical enzyme, the viral protease Mpro. This enzyme plays a crucial role in processing the proteins that the virus needs to replicate within human cells. By blocking this enzyme, the drugs prevent the virus from effectively replicating and spreading. Unlike other treatments, these new compounds have shown no harmful interaction with human proteases, enzymes essential for normal human bodily functions. This specificity offers a unique advantage. The likelihood of serious side effects, such as organ damage or other health complications, is significantly reduced. Early testing, including trials in mice, has suggested that these compounds could be not only effective but also safe, raising hopes for future human applications. What sets these drugs apart from existing treatments, such as Paxlovid, is not just their efficacy, but their broad-spectrum potential. While Paxlovid was primarily designed to target SARS-CoV-2, these new compounds have demonstrated activity against multiple coronaviruses, including MERS. This is especially important because the risk of future pandemics caused by coronaviruses remains high, and having a class of drugs that can target multiple strains of the virus could be an invaluable tool in pandemic preparedness. The ability to modify and manufacture these compounds easily adds another layer of optimism, as they could be quickly produced and distributed should a new viral outbreak occur. The speed and efficiency with which these new antiviral candidates were developed highlight the power of collaboration and innovation. Within just 3 years, the UCSF team, working alongside researchers at the Gladstone Institutes and a network of international collaborators, were able to advance from virtual molecular docking studies to real-world drug candidates ready for further testing. This rapid pace of discovery, often characteristic of the pharmaceutical industry, was achieved in an academic setting, underscoring the critical role of academic research institutions in global health innovation. However, despite the incredible promise of these new drug candidates, their future is uncertain due to a sudden and unfortunate termination of funding. The National Institute of Allergy and Infectious Diseases, NIAID, which have been supporting the research, cut its funding to the project, leaving these potentially game-changing drugs in limbo. The loss of this funding is a stark reminder of the fragile nature of scientific progress, particularly when it comes to long-term preparedness for pandemics. Pharmaceutical companies, often driven by short-term profits, have largely abandoned the development of drugs for pandemics that might not emerge for years or even decades. This makes public funding for such research even more critical, as it allows scientists to focus on preparedness without the pressure of immediate financial returns. The fact that such an important line of research is at risk due to funding cuts is both frustrating and concerning. With COVID-19 still causing global disruptions and new strains continuing to emerge, the need for effective antiviral treatments is undeniable. Yet, without continued financial support, the promising progress made by the UCSF team may be halted, delaying the development of treatments that could save countless lives in the future. For the sake of global health, it is imperative that the necessary resources be allocated to ensure these compounds can be tested in human clinical trials and, if proven effective, brought to market. Beyond the immediate benefits of treating COVID-19, the implications of this research extend to broader public health concerns. The novel compounds developed by UCSF and the Gladstone Institutes represent more than just a solution to an existing problem. They're a blueprint for how we might respond to future pandemics, particularly those caused by coronaviruses. The current research has illuminated the importance of creating antiviral drugs that are not only effective against a specific virus, but are also adaptable and scalable for use in different situations. The ability to quickly modify these drugs for new variants of the virus or entirely new strains gives us a level of preparedness that has been sorely lacking in the past. Looking ahead, the development of these antiviral compounds provides a glimpse of hope in our ongoing battle against infectious diseases. They represent the kind of forward-thinking, proactive approach to global health that is needed in a world where pandemics can emerge suddenly and without warning. The promising results of the UCSF team's work signal that the next generation of antiviral therapies is within our reach, and if we continue to invest in these efforts, we may be better equipped to tackle future health crises more effectively and efficiently. The new antiviral drugs from UCSF are not just a testament to the dedication and expertise of the researchers involved. They're a symbol of what can be achieved when we prioritize global health and pandemic preparedness. As we move forward, it is crucial that we continue to support research and innovation in this area, ensuring that the breakthroughs made today can be translated into real-world treatment for tomorrow. With the right support, these antiviral compounds could be a game changer for COVID-19 and for the future of pandemic response, potentially saving millions of lives and reducing the impact of future outbreaks.